you everyone for coming. I'm delighted to actually have this first session of our Biophotonics workshop, which is now open to the public. And today we will talk about the big picture of biophotonics. So very key concepts we will use throughout the workshop. And I want to convince you today that biophotonics is actually widely present in our daily lives. It has applications in both micro and macro scales. It is widely used for disease detection and treatment, and it has potential to revolutionize healthcare. So one of the most successful technologies of biophotonics is pulse oximetry. You may have had contact with this technology before when you have gone to a hospital and a physician just puts a clip in your finger. And what they do is actually they monitor your blood oxygenation and heart rate. But that is actually applied in several other places. So you can see portable pulse oximeters that you can connect to your smartphone. You see this technology in smartwatches. So when you are playing sports, when you want to monitor the quality of your sleep, you can look at the blood oxygenation and heart rate. And actually just talking about sleep, Netflix actually proposed to put pulse oximetry in socks. So when you are wearing socks and you just fell asleep during that very interesting movie, then when you fell asleep, the TV will just detect when you sleep and it will switch off automatically. So when you come back to that interesting movie, you have it in the same place as when you slept. So this technology is actually facilitating everyone's lives, and it is based on actually using two colors of light to detect the absorption of oxygenated and deoxygenated blood that is in your finger or in your wrist if you are wearing a smartwatch. And typically in medical devices, we use a red light and a near infrared light to probe the oxygenated and deoxygenated blood. So what we do is, depending on the proportion, we have a certain absorption and we can determine the oxygenation from that. If we are looking at the signals over time, we can actually look at when it rises, so we know when the heart is beating. So that's how we determine heart rate. But we don't only see blood signals inside the body, but we also see it outside. So you can use microscopy, for example, in forensic microscopy, to analyze the blood in a crime scene. And you can actually see many details of hairs and other objects in the scene just with this same technique. Microscopy is also used in several biofluids, not only blood. So for example, urine, you could see several of the laboratory tests, and urine is actually used to investigate if you have all these list of diseases, infections, and even cancer. You would look for several crystals or viruses, microorganisms, and so on. So you could see all this in our urine sample. Now, if you want to actually get rid of infections, if you want to disinfect and decontaminate surfaces, you would actually use ultraviolet light. And this technology has been used in hospital rooms for several decades already. And it is also used in food industry to decontaminate the surface of food, which would be a nightmare to clean, right? The disinfection decontamination technology has actually been booming since the pandemic. Now that we need to actually decontaminate and disinfect everything, as long as it's in a crowded place or whatever we need to decontaminate. So biophotonics has been taking a huge role on this type of application. And biophotonics is also successful in imaging inside the body. So we could use a scope to look into the gastrointestinal tract. We could look into ulcerations, infections, tumors, and so on, with several procedures such as endoscopy and colonoscopy. And biophotonics can be used also to guide surgeries. In this case, the light-based technologies would facilitate to the determination of where the surgeons can cut or can actually remove tissue or even identify tissues that shouldn't be removed. So, for example, if you could have important blood vessels or nerves that shouldn't be removed in the during the surgery because the patient actually needs that. And if you remove an important nerve, you could cause paralysis, or it could have a lot of huge consequences. But biophotonics is not only used for imaging and identifying the location and the type of disease, but it's also used for treating them. So with the advent of laser, we had a lot of laser surgery procedures, and one of them is, for example, eye surgery, which one of these types is LASIK, so the laser-assisted in situ keratomyelitis. In this case, we would have we would be able to correct the eyesight of a person by doing this type of surgery. And just in US, there are actually over 700,000 procedures performed per year. And this is a decrease from the past years because this procedure doesn't need to be performed again for a long time after you have it. So this procedure is actually booming even more in countries under development, and it is also an important procedure that was enabled by biophotonics technologies. Now, there are other procedures such as lithotripsy, which consists of breaking kidney stones, which are actually blocking the passage of urine from your kidney to your bladder. And this actually causes pain and nausea in patients, and it is a very discomfortable process. In a lifetime, 13% of all men will have this accumulation of kidney stones, and 7% of all women will do as well. And once you have this, you have 50% of chance of having this accumulation again after 5 years, and 80% of chance to have it after 10 years. So not only almost 10% of the population have this and needs to go through the procedure, but we actually see that biophotonics has been successful in solving many problems in the society. Now, these are all procedures where a lot of a very intense light is applied in a very small object, a very small volume. But there are several treatments which also use light spread out over a larger volume. And in this case, we would have light therapy, which would modulate the metabolism of several tissues. We call this photobiomodulation here. Photobiomodulation is used for many applications, including aesthetics. So for example, acne and wound healing. Here you see that the results are after photobiomodulation are amazing. This light therapy is also used in sports medicine to improve the performance of athletes and even healing their injuries. It is also used for pain relief and anti-inflammatory response based on vasodilation, for example. And photobiomodulation has also many applications in diseases and disorders. This is a map of the disorders that are just treated with photobiomodulation. As you can see, it's a very extensive list, and you can treat diseases all over the body. And in photobiomodulation, you would treat them by having the metabolism of the tissue modulated in the location where you illuminate the tissue. Now, if you want to be even more specific, and in the area you illuminated, you want to target only specific cells, what you should do is actually you make those cells more sensitive to light so that they will also have more predominantly the effect. So this is done in one of the procedures called Photodynamic Therapy. In this case, the cells are actually they become more sensitive to light, and upon certain conditions that are met, the cells which were photosensitized or which are more sensitive to light will be killed selectively. You can actually kill cells of fungi or cancer cells selectively using this type of treatment. And here I just show you a list of the human infections which are clinically treated with photodynamic therapy, meaning these infections are already, we can treat these infections daily in the clinic. It is a procedure that is performed all the time and already approved. But actually photodynamic therapy is used for many other diseases other than infections. It is researched for cancer therapy to many other types of applications. And just to give you an example of what it looks like. So if we see a fungal infection in a nail, this is what it is before, it looks before, and after the treatment, we can completely get rid of the fungi that are infecting this nail. So the results are amazing, and there are several successful technologies, not only with photodynamic therapy, but we can also see one of the common technologies with the retinal exam. So the retina will be this layer in the back of your eye, which is used to actually detect light from the external environment. And retina can have a lot of irregularities depending on the disease you have. So you can use a technique called optical coherence tomography to look at these irregularities. And you may not be familiar with this name, but I'm sure you have seen an equipment like this when you went to the doctor and they just told you to place your chin and your forehead in the equipment and look at an object very far away. So if you don't move your eye, what they will be looking at is the profile of your retina. So they can look for several diseases. It could be ocular diseases, could be even indirectly related to diseases such as diabetes. So many of the successful technologies are out there, and this you can see even in the medical imaging market for biophotonics that optical technologies or light-based technologies are actually very predominant. Actually, most people, when they refer to medical imaging, most people actually have in mind X-rays or ultrasound images or magnetic resonance imaging, or actually if you have any relatives with cancer, you might think about radiotherapy. But actually, the market for the radiological technologies is much smaller than the optical. The light-based technologies actually have almost double size of the market compared to the radiological. And this is not including home healthcare. If you count, this is even more prominent if you count the mobile health, which in 2016, before the pandemic, was about 13.2 billion, and wearable devices, which was 5.5 billion. So after the pandemic, needless to say, but everyone is at home, home healthcare is way more important, right? So then the market expanded quite a lot in the optical side, especially because we can't use, for example, X-rays at home, right? So we have this growth of biophotonics in the past years, and let's try to understand why biophotonics is so successful. So just quickly remembering a bit of the basics, if you look at the electromagnetic spectrum, you might remember that a wavelength is the minimum distance where you have the repetition of a wave shape. And once you increase this wavelength, then you decrease the energy of the light, and the visible light so the light we see is in this very narrow range here, which goes from 400 nanometers in violet. It goes through blue, green, yellow, orange, red in 700 nanometers. And actually the electromagnetic spectrum goes much over than the visible range. If we observe this spectrum, we actually have the highest transmission for water in 3 regions which are used for medical imaging. These are the X-ray range, the optical imaging range, and the magnetic resonance imaging range. And the optical range is actually the one with highest transmission for water. And not only this, optical, most of the information of most of the useful molecular information is actually optical. Now, if you think about how several changes occur in certain diseases, you can think how these diseases will develop within the cell. First, there is some mutation, some genetic mutation, and a genetic material such as DNA is actually going to lead to the production of proteins due to this mutation. And this will culminate in several changes, several immunologic changes, which will happen in molecules which are present in a higher concentration compared to just the genetic material. If the immunological changes are persists, then we can have a lot of metabolic changes, which will again have a higher concentration of molecules, and it will be easier to be detected. And if the metabolism is changed for a long time, we will have then the structural changes, which are targeted by conventional techniques we know today. So structural changes are very easy to detect, while changes going towards genetic changes would actually be more specific, because then we actually see several parts of the molecules which are very specific to certain diseases. Whereas in structural changes, we could have any cause for that change. Now, if we think about conventional technologies such as X-rays, MRI, and so on, they probe mostly structural and metabolic changes. Optical technologies can actually detect diseases much earlier than that and much more specifically. So because we target the immunologic window there. And we can also have a variety of diseases that could be probed with optical technologies. We actually have a number of molecules we can detect. Now, thinking about all these advantages, let's go through some of the key topics in biophotonics. These would be first the fluorescence and microscopy, and we go through the other topics right after. So now if we think about just a conventional microscopy, what you have is a very straightforward path with light illuminating the sample from the bottom. You would see that the light passes through a condenser lens, which will focus the light in a plane within the sample. And that image will then be magnified by an objective lens and an ocular lens before going to your eye. So you see that very big image of something very small. But microscopy can actually be combined with fluorescence to enhance the images you see. And in this case, you can observe many types of biological processes, such as cell division. In this case, I'm showing you a GIF here, where you can see the processes of the steps of cell division. And you can see the nucleus disappears first, and then you have this DNA going to the center, and this is pulled out to the edges of the cell, and the cell actually divides into daughter cells. And you see that this image enhancement of fluorescence microscopy is actually makes the image much better than conventional microscopy, where you cannot see many details. And this enhancement is actually due to several advantages of fluorescence. Usually we have more sensitivity, so we can detect smaller changes. We have more contrast. Usually in fluorescence, we try to have measurements where we don't have background light. And we also can label multiple cellular structures at a time, so that we can see several processes in parallel. And we can, to label these structures, we can actually use fluorescent dyes at different colors. So we can see the biomolecular processes in, for example, inside the cell by labeling certain proteins, which have specific locations. We can also use fluorescent dyes to label certain cells in a mouse brain and see where they are located. We can also probe several organs inside the organisms and even an entire nervous system of a zebrafish. This was only possible because of the discovery of the green fluorescent protein. And this discovery actually gave these three people their Nobel Prize in 2008. What they did is actually they discovered this green fluorescent protein in jellyfish, and they saw which of the DNA structure would generate this kind of protein. What they did is then they modified the DNA of a certain organism and saw if this organism would produce the GFP. And upon there was, they generated a fluorescent organ, for example. And this actually revolutionized many of the ways we can look at biomolecular processes and how we actually see that's happening in both macro and micro scales. Now, we also see in macro scales a lot of advances in precise localization and single molecule information. So, for example, if we use super resolution microscopy, we can actually probe the nuclear pores, so the pores within the nuclear envelope of the cell nucleus. Now, if we see this image on the right side, you see that with confocal microscopy, which means a type of microscopy where we get only light from the focus, we cannot actually see anything. But if we use super resolution microscopy, we can actually see the structure of the pores, which is like this, with the basket and the ring really well defined. So why we can't actually see anything with just focusing light in a point? We actually are restricted by the diffraction limit of light, and this doesn't allow us to focus light in a smaller distance than a wavelength of light. So how could we actually make super resolution microscopy if even the best equipment focusing light couldn't actually do it? So the people who discovered how to do that actually won again a Nobel Prize in 2014, and the submodality of super resolution microscopy is the super result fluorescence microscopy. One of submodality of even this is the stimulated emission depletion microscopy, which I'll explain you here. So imagine just a confocal setting where you have the light being focused in a certain part of the sample. What we want is to identify where the location of these two beads here. These beads are separated by a wavelength of distance of a wavelength. And what we do is that when we scan these beads with just focusing light in a point, is actually we see a rising intensity, and once we pass through the beads, we just see the dip there with a decreasing intensity. So we can't really see two beads, we can see only one, just a blob. But what the Nobel laureates thought is that, what if we create then a donut, if we can't focus the lights in a smaller region, if we do a donut, and actually this structure will have just a hole in the middle, which is actually smaller than a wavelength. And what they do is that they focus light using a donut, and in the area of this donut, there is no fluorescence because there's this emission depletion that we just have in this type of technique. So in this case, what happens is that when the bead passes through the center of the donut, we have the emission of fluorescence. And we will have then this emission only when the beads pass through that. And it happens in a much lower scale than a wavelength. And that's how we do super resolution microscopy. Now, we don't only see small things, but we can manipulate these things. So we can actually do optical micromanipulation, and Arthur Ashe can actually use optical tweezers to do that in biological systems. And because of this, he was a Nobel laureate in 2018. So this is based on a principle of optical trap, and certain structures will be going to the focus of a laser beam when we do that, when we focus it there. So we can use that effect to actually concentrate red blood cells in the illuminated region, for example. We could actually do several machines to control, micro machines to control the flow within cells. And there are many other applications we can do to study mechanical properties and other things inside the cell. So if we think about both micro and macro scale things, we can do some imaging in both scales at the same time. We use, in that case, optical clearing methods. And you can see here in the left side, for example, the image of a mouse, which before it is cleared, and after it is cleared, you see that it becomes transparent. And with this, we can actually image an entire skull of the mouse. We can image entire brains with it. So you could see just not only the image of the whole brain, but actually you see it in a very good resolution. You see all the nerves, you see all the cells, neurons, and all the details in there. So this is actually amazing, and I will show you actually a video about that.
Being able to observe a neutrophil just crawling through the ear of the zebrafish, scooping up all of these food particles that we've injected, that's just mind-blowing. I actually got goosebumps in my hair. I still have goosebumps thinking about the moment when we actually first saw that. Being able to visualize that was phenomenal.
Cancer cells. So these are human cancer cells that are being implanted in the zebrafish. And you can see now the cells moving along capillaries, cells crossing the wall of the capillaries. But it's not just a blob of the cell like other people have seen in the past. This time we see the details of how the cell is sending pieces of the cell to explore their space.
We've mapped the entire eye of the zebrafish and then computationally exploded the cells such that you can look at each cell as a unit. And what you can actually observe is the changes in morphology in the context of the tissue. That's never been done before. You can look at how all of the intercellular compartments are dynamically changing. Right? We can see cell mitosis, cell division, cells kind of pushing against, beating up their neighbors while they're kind of violently dividing, or at least that's the impression I get when I'm looking at the cell divisions.
Now I cannot sleep anymore. I'm up every night trying to figure out how to do the next thing.
If we come back to our presentation, then we not only have microscopy, but we can also have optical techniques in a larger scale. So in the now in a certain in fluorescence disease diagnosis, where we probe tissues, biological tissues, we have two types of techniques. Mainly, we have those using contrast agents. In this case, we have increased sensitivity, so we can probe very small changes in tissue. And in this case, we have side effects of injecting fluorescent dyes, and we need to perform the procedure while the dyes are within the system. Still, this can be very good for very quick procedures, and it is the one used most widely. Another type of technique is label-free, so using no contrast agents. In this case, it is a procedure, completely non-invasive procedure, and the only drawback is that we take more time to process the captured signals. to extract the relevant biochemical information from the tissues. Now, these two will have advantages and disadvantages, and we will see first an example of one procedure using contrast agents. First here we will have the procedure which can be combined with photodynamic therapy we have seen before, and this procedure is mostly based on a substance called aminolevulinic acid, or short is ALA. This is a molecule which is already present in our body. It is used to produce protoporphyrin 9, and protoporphyrin 9 is actually used to produce the heme group, which is in the hemoglobin of your blood cells to bind oxygen, and then your blood cells will be able to distribute oxygen all over your body, so you can produce energy to survive. So ALA is very important for your organism, and in this type of procedure, what we do is that we can externally administer ALA to a person, and in this case, the ALA will induce the accumulation of protoporphyrin 9 over time. If we wait sufficiently, since protoporphyrin 9 is actually a fluorescent molecule, we can see that when we illuminate tumors, they are a fluorescent molecule which actually accumulates preferentially in tumors. And when we shine a blue laser in the tumor, we can see the fluorescence in red coming out of that tumor. What it looks in a diagnosis perspective is that we see the fluorescence intensity over wavelength, which is higher at 635 nanometers when we are probing the tumor. And we can look for that spectral signature to check for the cancer margins in that application. So when we actually have this type of identification of cancer margins, we can actually do treatment as well. In this case, since protoporphyrin 9 makes these cancer cells more sensitive to light, we can actually shine a red laser, and actually we can kill the laser cells with the cancer cells with the same procedure. We perform disease detection and therapy. Now, if we are diagnosing that, we actually look at the fluorescence spectrum of that. A normal tissue will have a fluorescence just from the normal molecules you would find in a tissue. And the tumor cells, in that case, will have this accumulation of protoporphyrin 9. You see the malignant tissue on something like that with the peak of 635 nanometers. Now, if we look at what's the difference in a procedure where we use contrast agents and where we don't use, let's take an example of a brain tumor. So if we take the tumor here and we look for the spectra at different locations of the tumor, so the spectra located out of the tumor will actually look normal, but if we look at the spectra on the border, we have this very pronounced peak of protoporphyrin 9, and the same inside the tumor. In a procedure where we have, in a label-free procedure here with autofluorescence, we have outside the tumor just a normal spectrum, and in the edges of the tumor, in the boundary of the tumor, you would have this peak of protoporphyrin 9 because it still accumulates preferentially in tumors, but it's not as pronounced as if you inject ALA in the person. Now, inside the tumor, the protoporphyrin 9 peak is also present, but even at an even lower intensity. So it is possible to detect changes. We just need to sometimes process a bit more the signals, and this will be done very quickly. Now, we can think about the types of techniques we could use to detect. diseases. In this case, we actually have point monitoring, so we can understand better what is happening with the tissue biochemically if we collect the whole spectrum. If we actually know what is already happening, we can select a wavelength and then do imaging within an area. So in that case, we'll monitor how is the signal of the whole area at the same time. And there are hybrid technologies of these two, as you can expect. In diffuse optics, we can compare what we expect from the behavior of linear optics and diffuse optics just to understand how it works. So in linear optics, we actually have the light coming in a line. So we actually have, if we have a transparent medium with just a refractive index and inside and outside, we can have a certain specular reflection then from when the light reaches the medium. If it goes inside, we can, depending on the angle and the refractive index, we can have total internal reflection. And if the light is transmitted, we can actually have a direct transmission. This is how we see it in linear optics. But in diffuse optics, we actually don't have anything like direct transmission. In diffuse optics, the light bounces over and over inside the medium before reaching the other side. And you can see it in this example of a cuvette. In a clear medium, you have the light going through directly, and in a turbid medium or diffuse medium, you have it totally going all over. You can see it better in this image. So this is the top view of a glass of water where we have a laser pointer going through, and we increase bit by bit the amount of light scattering. So you can see that once we increase the scattering, then actually light really becomes more and more diffuse. So less and less light will reach the other side of the glass. So with diffusion, then we can observe many phenomena in nature. So you see diffusion in clouds, you see diffusion even when you go to the beach for your holidays, maybe a long time ago. But I'm sure that you have observed already the beauty of diffuse optics in the beach. You just didn't notice that. Now, you can see actually diffusion everywhere. We are diffuse, many objects are diffuse, almost everything is diffuse. We don't see through them. So we actually can see a very easy example when we just put our hand just in front of a flashlight. And in that case, we see that we don't see our bones like an X-ray imaging, right? So the light doesn't go straight so that we can see the bones directly, but the light goes all over around the bones, and we see just the diffuse shadow of them, which is not really clear where they are located. Now, to see an example of the diffusion, let's go over to Sanatana Kunugulu, so the researcher, senior researcher in the Biophotonics group at IPIC and Tyndall. And I hand over to you, Sana.
Thank you, Marcelo. Thanks for the introducing the concept so well. How diffusion is different from X-rays. So here is a simple. I will be showing that with a simple example. Just before going to our demo, just what I want to mention is that we all want to see through our body. It's already happening where X-rays or MRI scans. But as you know, X-rays are carcinogenic in the sense they can mutate your genes and cause you cancer. But this field has been evolved during 70s and 80s, so on, and we can take right now whole body MRI and CT scans. But this is not possible in diffuse optics. Why? The simple example is if I put my hand, you see the light doesn't see through. X-rays, if you put X-ray, it can see through. On the other hand, if I put light, it's not see through. So how does the X-ray technology work? Or the CT scan work? As you can see in this particular slide, means you have object. Let's imagine these objects as different organs in your body, and you just start eliminating from different directions. So one direction when you see, it look like triangle, and but the actual cylinder is looking like a rectangle. And on the other direction, you see, again, the cylinder is going to look like a rectangle. When you back project them, then you will get the shape. Just to show an example in the hand. So I have this spoon here, right? When I see like this, it's flat. Okay, you remember that. And I turn it around, it's kind of straight. Then your mind projects all these things to reconstruct inside yourself. Oh, this is actually a kind of spatula that has flat and a thin surface. That's how the CT scan works. But we can't do that in diffuse optics for simple reason of very high scattering. I'm just going to show you through the example. Let's get to our demo table. Just let me know if you can see what I show here. Just give me a second. Okay, so to start the example, all the thing that you need is some milk, okay, and a tank and some water, okay, and just pour the water into the tank. It can be a tank or any tub, fish tub, things like that at home, okay? I'm just going to mount that there. Now we need a light source, right? So that we can do a projection. I'm just going to use my mobile phone LED, okay? So I'm just going to mount the LED here, okay? For now, you can imagine this as a kind of X-rays, okay? That's just going through here, and you have the water, and you have the back projection here. You can see it, right? Can everyone see that? I hope, yes. So I just filled up the tank. So imagine now no need to imagine, we are going to do everything here. So I chose the fork. I was having my lunch. Suddenly I thought maybe this is a good example. You see, your brain knows its fork because it rotates around and says, okay, it's flat this way and this way, it is this way. And eventually it creates the image of fork in your head. So I'm just going to drop the fork inside the fish tank with water. And you can see already clearly the projection of fork, right? That's what happens when you send X-rays. Now, just try to think, human body is made of around 60 to 70% of water. Why the human body is not see-through, right? That's the question that should come. Just remember, like most of the great inventions happened, when people are looking at nature. For example, Newton's law is nothing but when apple fell down the apple tree on Newton's head or whatever, he found the Newton's law. So these are the things that simple things that you can do in kitchen, but you can come up with great theories that will change the way we live in this world. Okay, getting back to the demo again. So you can see the spoon back projected. What I am going to do, these kind of X-rays, okay, you can see through, but our human body is not transparent like water, though we made of 70% of water. So why it is so, I'm just going to show you. I have milk in this particular syringe. It can be syringe or something just to calculate and drop. Let's see what happens when I drop the milk inside this water. Okay, just see, you can see even the fork, also the needles of the fork, right? Once I stop dropping some, now I'm dropping some, I hope you can see how I'm dropping the milk. I can use the fork to, just mix the milk as well. You see, the solution is getting turbid. And yeah, it might probably it's fading slightly. So you don't have the same contrast that you had before when you saw the fork before. So you are losing a little bit of back projection. Let's see what happens if I add few more drops of milk. Okay, now I'm mixing it. You can see, right? It's getting even more turbid, which means we can't see through the fork, not so great as we used to see before. Let's see what's going to happen if I add even more milk, okay? It's step by step. Just keep thinking in your head, why this is happening? Why this is happening? Means I'm just trying to convert this pure transparent water into a turbid medium, how our human body is, and show you like why we can't see through. And you see, you are seeing hardly, maybe some part of fork, maybe. Actually, yeah, it's not so much. There is a final chance. After this, you will see nothing, in my opinion. We just added some more milk. Now you can't even see anything. So this is exactly what happens in our human body. As we all know, with a simple Googling, human body is made of 60% of water. But as this actual transparent water is contaminated or by the milk, so milk has microscopic structures that is not that is scattering light, that is not able to, that is making this fish tank no more see-through. Similarly, in human body, though we are made of 60% of water, we have cells. In cells, we have organelles and mitochondria and so on and so forth. These are very, very microscopic structures like milk, which actually scatters the light that is getting through our body. So just to show you here, and it diffuses it out. That's the reason why we can't see through. Basically, we can't see through. We can't see our bones that we are like how we can see in X-ray. So this is basically a great challenge, right? But actual inventions happen, or the great things happen when you have real challenge and when you know good answers for it. So that's what we do in diffuse optics, or that's what we do in biophotonics. We are trying to understand how these photons propagate inside the diffusive medium, like human tissue, and recreate the path that the photons have taken so that we can see through human body once we reconstruct them by using some diffusion models. I wouldn't say it's that complex, but that is the approach that we have to do. So what is the summary of this whole thing? Human body, though it's made of water, it's not transparent simply because we have these cells and organelles that is scattering light. That means we can never see through the body? No. It's simply that we need to understand how the light propagates in this particular medium. By using simple visible light, which we normally see in every day, including this room. With this, Marcelo, I want to leave it back to you.
That's perfect, Sana. Thank you very much for the demonstration. That was amazing. So, very good. Bringing back your slide here, and let's just continue on diffuse optics then. So, now, after Sana's demo, you have understood that diffuse optics is actually, it happens all over the body. It's actually seen really, really through if you want to take the challenge and create a technology where we can determine all the locations of structures inside tissues. Please help us. We'll be delighted to hear from you. So now coming back to tissue optics then, let's think about same schematic as we had for the linear optics. When light reaches the tissues, then we can have specular reflection as in transparent media. But when it goes inside, actually light is totally scattered all over, and it can even come back to the same surface where you illuminated the tissue. In this case, we will be looking through the scattering happening there. And there are many other processes which can happen, such as absorption, or the light can actually be absorbed and emit light in a different color compared to that you used to illuminate the tissue. And that you see in fluorescence, phosphorescence, Raman scattering, and so on. So, and the light can also be scattered and transmitted through the tissue. Now, let's think about how this can actually be used to determine certain tissue biochemistry and structure properties. So here we'll be looking at the absorption of the tissue, and we'll be looking at the wavelength dependence of the absorption coefficient. A typical spectrum, absorption spectrum from tissues is like this. So we have a lot of absorption from blood and water. And in this case, what we can think about to understand this absorption coefficient is that we can take it as the distance necessary for you to see an absorption event. So if we have 1 centimeters minus 1 of this coefficient, you would have 1 absorption event after a centimeter. And this attenuates the light, obviously. And the region, the wavelength range where you have less attenuation is called optical window. This is where we are most transparent. So if you think about how much attenuation we have due to the absorption, then you can think about the Beer-Lambert's law. So this is to calculate how much light is how much attenuation we have in the intensity of light after it passes through a certain volume of tissue. So we can calculate this attenuation by this exponential of absorption times length. In this case, just think about 1 centimeter of tissue, and if you just take the absorption from the blue at 400 nanometers and red at 700, you can see that we have almost 1,000 times more absorption in the blue region. Now, if you want to know the attenuation intensity relative of the blue relative to red, you actually calculate that with the exponential just above. And you can see that if you have just 1 unit detected in red, you'd have a decimal of 400 zeros detected in blue. So it is a huge attenuation. And that's why the blue light will not pass through your fingers, but red light will. And actually, you would also consider scattering events, which will happen in this order here of the centimeters -1 with scattering coefficients. Now, if you think about the penetration of light in tissues, as you can see, the wavelength actually matters. And actually, you can see it not only by shining one type of light, but if you shine blue light, then you see that nothing passes through your finger, but red light does. And that's why we use red light in pulse oximeters to actually see the difference in oxygenated and deoxygenated blood. Now, there's also the reason why we use red light to reach blood vessels in the skin, for example, for having photobiomodulation and anti-inflammatory responses based on vasodilation. And thinking about this penetration, then we have how to actually probe for several tissue constituents in many medical applications. So we can not only look at the tissue constitution in terms of blood and water, we can also look at the constitution in terms of lipids, collagen, tyrosine, thyroglobulin, and so on. What we do is that we have the same absorption spectra as you saw before, and we can fit that spectrum to have the concentration of these molecules. And this can be done then in several applications. We can have the biochemical profile of the tissues in all these. At the same time, we can identify the location of certain parts of the tissue. So tumors, for example. And this is used in applications all over the body. So including thyroid, breast, bone, abdomen, head and neck, gastrointestinal tract, in many other parts of the body. So that you have a lot of applications with that, including disease detection, surgical guidance, and functional endoscopy and colonoscopy. Just to give you an example of that, in breast cancer diagnosis, we can have a tumor in the very center of the breast, and you see with X-rays that very clearly that that tumor is in there. And if you look at the tissue constituents, just with light, without ionizing radiation, just from light outside, you can see that the tissue around the tumor has a very different biochemical profile compared to the cancer. You see it in oxy and deoxyhemoglobin, lipid, water, and collagen. So you can see this profile directly. And there's just one application. There is an enormous list of them. And tomorrow you see more of those applications and even more in the Biophotonics projects at IPIC and Tyndall in the webinar, which will be given by Professor Stefan Anderson Engels. So don't miss that. I'm sure you will be very excited about what we do in IPIC and Tyndall. So now, very good. We also use the information of absorption of specific constituents to propose several treatments to diseases. So an example of those are hyperbilirubinemia and jaundice, which happen in 60% of the babies born, so a good fraction of them. And in these diseases, we have accumulation of bilirubin, which is a yellow pigment. And this pigment actually is generated by the breakdown of red blood cells. And when it's not removed, it accumulates in several organs, such as the skin, and the baby is totally yellow after that. You can see that bilirubin actually has a blue absorption, and that's why we actually can break bilirubin by illuminating the baby with blue light. And I will show you a video about that. See this screen?
Jaundice is a common condition in babies. It presents as a yellow discoloration of the skin. This yellow discoloration is a buildup of bilirubin. Some babies with high levels of bilirubin will require treatment. The most effective treatment for babies with jaundice is phototherapy. Phototherapy involves shining light both below and above the baby, and this light changes the bilirubin from being dissolved in fat in the body to being dissolved in water, and therefore excreted in the urine. Babies admitted to the intensive care unit requiring treatment for jaundice will require blood tests to ensure the bilirubin level falls, and as these levels fall, the lights are then removed from the baby. When the level is below the line that is requiring treatment, the baby will be able to be discharged home. Phototherapy is an effective treatment for jaundice.
So this is how we help all the babies all over, many babies around the world, just to actually remove their bilirubin from their bodies. Oh.
Hi.
Yes, so we can also use the specific absorption from each of the constituents, but even the external constituents that we can induce cells to produce, to actually induce, stimulate, and inhibit certain types of cells to behave how we want. So for example, this is used in optogenetics to stimulate and inhibit certain types of neurons. Usually in electrical stimulation and light stimulation, we will stimulate or inhibit all the neurons in a certain region where we had this electrical stimulation or light stimulation. But if we want to select a few types of neurons to stimulate or inhibit, we can actually have them producing a certain substance which will absorb a certain a specific type of light there, and they will do they will generate a chain of reactions which will end up in a change in behavior of the organism. So here there is a video. I will show you how it is. So what happens is that when light is switched on, the rat or mouse there will have the sensation of thirst. When it's switched off, it will just move around as something else, as nothing is happening. And when it's switched on, it will have the sensation of thirst again. So that's how we can stimulate or inhibit several types of neurons in order to stimulate certain behaviors in organisms, and even having a studying certain diseases, and even studying how the brain works and how the connections are made between certain types of cells. So optogenetics is actually a very good optical technology. It is really impressive, and we will see its prospects in the coming years. Now, if we talk about future trends in biophotonics technologies, we actually can think about first what would be the dream of biophotonics. In biophotonics, we actually have the dream of looking like Star Trek. So if you have seen Star Trek, you might have seen the tricorder, which is just an equipment which can scan diseases all over your body at one go. So this is what we want in biophotonics. And we are moving towards that. We can actually combine optical and ultrasound technologies to actually get the advantages of both. So light, optical technologies, we will have actually a very good molecular contrast. We will be looking at the biochemical profiles really well, but we don't have so much resolution at deep layers of tissue. Now, if we look at ultrasound, ultrasound doesn't have molecular sensitivity, but it looks at very deep tissues. We can combine the advantages of these two, so we can have biochemical profiles of deep tissues. And one way of doing that is using acoustical tomography. In this case, we have a laser that is shining light in the focus of an ultrasound that is passing through the tissue. And the detector will then detect primarily the laser light, but also will detect a frequency shifted light, which can be tagged. And if we detect only this type of light, we filter it, we can actually image tissues of over 18 centimeters deep, which means we can image through the body. And if we image throughout the body, we actually are doing the same as the tricorder, right? We are moving towards Star Trek, or at least the tricorder. So yes, we can use several technologies to also look at how we can scan tissues even superficially with optics. So one way of doing that is during surgery. With augmented reality, which is now becoming more and more popular, especially with the VR and AR headsets, the surgeon can actually wear one of these headsets and probe certain regions of the tissue and look for very specific spectral signatures or biochemical profiles related to that. And it is possible to then find the cancer margins with this technology. This is a technology that is upcoming in already for maybe 2 years or so. And it is going to, I believe it is going to reach the market soon. So we will see some of those now. You will not use your VR and AR headsets only for games. You use it for medicine. So now thinking about this way of having biochemical profiles of tissues, we can also have biochemical profiles of very small structures such as coronary arteries. And in this case, we can image inside and have profiles of collagen, elastin, and lipids with inside these blood vessels. So this is with a very tiny sensor, we could actually have even detection in real time with all of that, and detecting diseases in very small structures. So for this, we need obviously miniaturized instruments, and a set of miniaturized instruments have been already developed. We do a lot of this already in IPIC and Tyndall. And examples of those are already present in wearable devices, such as maybe your smartwatch, if you have one. It is also present in medical devices, such as endoscopes. It is integrated in the tip of it. Even in catheters, as you saw in the previous example. Miniaturized instrumentation is also present in microfluidic chips, which can be used then to analyze biofluids. Could be blood, could be urine, saliva, and so on. So these chips can also be sent to many places. You can even, if they don't have the facilities to actually have a lot of biochemical analysis of samples, they can do it with the chips. And this is also very important with the pandemic, right? Because now we actually don't have a lot of access to so many facilities, and this can actually be used to have technologies spread elsewhere. So miniaturized technologies are also coupled to smartphones. You can have spectrometers coupled with your smartphone. You could have microscopes and other instruments that you can actually count the number of cells, which from from a diseased sample or even a biofluid sample that you, with just a module that you can couple in your smartphone. This has already been developed for, yeah, maybe over a decade, and this is more and more coming to the market now. Now you can actually detect diseases with just a smartphone, and we are even having more apps for that. All these technologies are actually contributing to the telemedicine, and the telemedicine has as well grown so much after the pandemic. Now, the doctors will be able to have recommendations to their patients from a distance. And if we can monitor the health signals of everyone from a distance, it will be much easier for the doctors to actually give recommendations for all the patients in a good way. We have more details and we have better health prospects with that. And especially if people are at home, this is very important. Now, I hope I have convinced you that biophotonics is widely present in our daily lives. It has applications in both micro and macro scales. It is widely used for disease detection and treatment, and it has potential to revolutionize healthcare. Here I have some of the references of images used in the slides and the YouTube videos, and also I leave here by the slide with my content.